Adopting breath tests for the diagnosis of SIBO has facilitated a better understanding of the role of SIBO in conditions such as IBS, celiac disease, and others. However, an ongoing criticism of breath testing is the lack of studies comparing breath testing to the small bowel microbiome. Evidence shows that breath methane levels correlate with a microbe called Methanobrevibacter smithii in stool and also with constipation. On the other hand, breath hydrogen sulfide levels are linked to hydrogen sulfide producers and diarrhea. But how do these gases relate to microbes in the small bowel? In this study, we hypothesized that breath methane and hydrogen sulfide levels would also be reflective of, or influenced by, small bowel microbial populations. Reimagined study subjects did a fasting single-sample breath test to measure their methane and hydrogen sulfide levels. The same day, subjects had another endoscopy and researchers collected fluid from their small intestine. Microbial DNA was extracted and shotgun sequencing was performed. Subjects with higher breath hydrogen sulfide, 1.5 parts per million or higher, had different bacteria in their small intestine. They had more microbes from phylum Thermodesulfobacteriota and more hydrogen sulfide producers like these three organisms. Subjects with all three of these species had hydrogen sulfide levels about 50% higher than those without all three of these species. Subjects with intestinal methanogen overgrowth, defined as methane levels 10 parts per million or higher, had more methanogens, including these four species. EMO was more common in subjects with all four of these species in their small intestine. In those without EMO, P. mirabilis and D. oligotrophicus were linked to looser stools. This study demonstrates that breath test findings were related to the levels of small bowel microbes producing these gases. This reinforces the importance of breath testing and its interpretation. Identifying the organisms responsible for breath test gases, their precise gut locations, and their consequences for human health will help develop better treatment options for SIBO, EMO, and hydrogen sulfide overproduction.